Grand Rounds this Thursday morning. Today we are joined by Dr. Presti and Dr. O'Halloran. Dr. Presti is Associate Professor in the Department of Medicine, Division of Infectious Disease, and the Medical Director of the Infectious Diseases Clinical Research Unit. Dr. O'Halloran is Assistant Professor in Medicine in the Department of Medicine, Division of Infectious Diseases. She additionally serves as Associate Medical Director of the HIV Virology Clinic and is an investigator in the Infectious Disease Clinical Research Unit. This morning, they will be discussing updates on clinical research on COVID-19. Thank you to you both for being here this morning.
Thank you very much. Well, I could. Okay, so we don't have any disclosures, although we are doing all of these studies. So today we're going to talk a little bit, and most of this is going to be review for people, a little overview of COVID-19 disease, and then a little bit more on immunity to COVID-19, because a lot of what we're talking about is going to involve immune response. And I'm going to give a little bit of an outline about Operation Warp Speed and the NIH trials that we are doing, and how they are sponsored, and how they are being coordinated. And then I'll talk about active immunity, really in the context of vaccination against COVID-19 and the vaccine studies that are ongoing. And then talk about passive immunity, or transfer of antibodies, and outpatient studies that we're doing in that arena. And then we'll switch, and Dr. O'Halloran will talk about immune pathology and treatment of inpatients, and then a bit about the observational cohorts that we have put together and future directions. Okay. All right, so you all know this. The virus we're talking about is SARS-CoV-2, the severe acute respiratory syndrome coronavirus 2, and then the disease is COVID-19, coronavirus disease 2019. So coronaviruses are large enveloped positive strand RNA viruses that can be divided into 4 genera. And the picture there on the right is a model of coronavirus, and is actually on all of the slides, just to remind you. Jane is going to talk about this quite a bit more lately, but just to remind you, this virus is somewhat different from other pandemic respiratory viruses, including influenza, in that there is a pre-symptomatic phase after infection, and then a viral phase where there's quite a bit of viral replication, and you start seeing symptoms. We'll talk about those a little bit, but those are respiratory viral symptoms, plus a few slightly more unusual symptoms, including gastrointestinal symptoms, as well as, in some people, a loss of taste and smell. And then when the virus starts to be cleared by the immune system, in some people there is this cytokine storm phase where there's quite a bit of immune response, and some of the pathology at that point, particularly in hospitalized patients, is potentially more due to the immune response than it is due to viral replication. During that time, you start to see a hypercoagulability, and we start to see evidence of that clinically as well. And then there's a late hyperinflammatory phase that lasts out to a month. At this point, there's very little viral replication, and the pathology is mainly immune pathology. So just to go over the basic sort of demographics, and this is a little old, but the numbers haven't changed except that they are going up again currently. This is from the St. Louis County statistics, and they changed their graph, so I couldn't find the updated graph. But this is mainly just to demonstrate, in particular, that the death rate is not uniform across people who are being infected. In that we are seeing a much higher rate of death in Black or African Americans, not just in St. Louis, but around the country, and then an extraordinarily higher rate of death that is related to age. So as you get into the older populations, the rate of death from COVID-19 increases, you can see almost exponentially. So, and this is more updated from COVID-19 and throughout the United States, looking at hospitalization rates, and they're broken down by age. And you can see here that it's really the people over the age of 65 that are seeing the highest rate of death or hospitalization as well as death from COVID-19. And we're starting to see those numbers rise again here as we enter into the fall. Just to consider diagnosis, and I sort of mentioned this before symptom onset, it's less likely that people are being detected except in screening programs because most people are not being tested until they actually develop symptoms. But there is viral replication prior to symptom onset, and there is transmission prior to symptom onset, which is part of what is making this virus difficult to control. And then once symptoms are begun, that's when you actually see the highest rate of PCR positivity. And then after a week or two of disease, but maybe only a week and a half or so of symptoms, we can start to see antibody. This is actually important because this gives us the window of when giving exogenous antibody may actually be beneficial. So once someone starts developing their own antibodies against SARS-CoV-2, we see a decrease in the response to the administration of exogenous antibodies, either via convalescent plasma or the monoclonal antibodies that we'll talk about later. You can continue to see virus in multiple sites, including the nasopharyngeal tract, the respiratory tract, in bronchoalveolar lavage, so deep in the lung, as well as stool. It is less clear how much virus that's being shed in the stool is actually contributing to transmission, but it is detectable for quite some time. And then you can see, as is true for most antibody responses, there's an initial IgM response and then an IgG response to the virus. So again, I showed you before the picture of the virus. This is just to emphasize that most of what we're talking about in terms of therapeutics in both the vaccine and the monoclonal antibodies are therapies that are directed against the spike protein. And the spike protein is that blue thing that's sticking out on the outside, or in the pictures that are at the bottom of the slide, the little red spikes that decorate the outside and really give the coronavirus its name of coronavirus. And then what actually seems to be particularly important, then shown there on the right, that purple area is the receptor binding domain. And so many of these antibodies are directed at that domain, which binds the ACE2 receptor that the virus uses for entry. So antibodies, there's been quite a bit done looking at antibodies by both age and disease severity. So we still don't completely understand how the antibody response works, how long it lasts, and how protective it is. There have been studies, including one study of patients with mild symptoms who had much higher antibody titers in older and middle-aged patients when compared to younger patients. I will say that recent data on some of the vaccines, including the Moderna vaccine, demonstrates that there's a good antibody response against the vaccine, even in older patients. So it's not as clear whether or not these higher levels of antibodies may be protective or may actually be an evidence of underlying more severe disease in the older population. There was another study that was recently published, and we actually have seen similar data with our own cohorts, that we see higher titers of immunoglobulin, of antibody, in patients who've had severe disease versus non-severe disease. So just for example, looking at people who've had high fevers with disease when compared to people who did not report fevers, even with mild illness, we see higher titers of antibody in those folks following resolution of disease. So there are other reports, though, that do not see these kinds of differences. So we're still trying to figure out what's going on with the antibody response. And then the million-dollar question is, how long does the antibody response last, and how protective is it against reinfection? There have been some limited reports of the possibility of reinfection. Most of those demonstrated a less severe disease on reinfection, although there have been people who've had more severe disease with reinfection. But it doesn't seem like we're seeing a lot of it, but again, it's too early really to say. So there has been a demonstration that antibodies against that spike protein in the animal models, so in non-human primate models, antibodies against the spike protein do provide protective immunity. And so that has been shown both in a re-challenge model. So that top slide is showing you there's a primary challenge, and you can see all the viral RNA that is detected after challenge in the non-human primate model. And then you can see once you allow those animals to recover and look at a re-challenge with the virus, you see very little replication of the virus at that point. And then you can see that's highly statistically significant that there is a difference in peak log of RNA between the primary challenge and re-challenge. Now, whether or not that is due to antibodies or to the entire immune response, including the T cell response, is not as clear in these studies, although neutralizing antibody titers correlate with protection against re-challenge in that model. More demonstrative that the antibodies may really be particularly important is shown in the figure below, where we look at vaccination with an adenovirus 26 vector that contains the spike protein. This is the vaccine that the Janssen study is using for vaccine studies in human trials at this point. This was the earlier data that was recently published showing that vaccination with that vector protects against challenge. And so for a sham vaccine, you can see high titers of virus on the left there, and then in the animals that were vaccinated against with the AD26 spike vaccine, you did not see viral replication. Okay, moving on to vaccine studies. I wanted to give a little bit of background in terms of how these studies are being conducted in the US and how they're being funded. So one of the major sort of drivers of the vaccine effort in the US is a program called Operation Warp Speed, which may be somewhat unfortunately named. But the goals of this program were to produce and deliver 300 million doses of safe and effective vaccines with the hope that initial doses would be available by January of 2021. That wouldn't be widespread distribution of vaccines at that point, but the initial doses would be available. We are still hoping that that will actually be the case, but as we'll talk about a little later, predictions are not always the way we want them to be. The idea behind Operation Warp Speed was to accelerate the development, manufacturing, and distribution of COVID-19 vaccines. So the program is not just to get a vaccine, but actually to be able to manufacture large amounts of it and to distribute it. And so that is part of the reason why we have multiple agencies involved in this. So critically important is the idea is to accelerate the development of vaccines as well as therapeutics and diagnostics, but to maintain all relevant standards for safety and efficacy. And so I'm hoping I can convince you that while we are doing this as quickly as we can, we are not sacrificing on standards of safety and not sacrificing the science. The Operation Warp Speed is managed by multiple agencies, including the Centers for Disease Control and Prevention. They are particularly involved in developing plans for distribution, particularly trying to focus on making sure that distribution of COVID-19 vaccines gets to the people that need them the most earliest. The Food and Drug Administration is clearly very, very involved in management of the studies and will be responsible for final oversight and approval of any vaccine that would become available. The National Institute of Health has been responsible for helping in terms of design of these studies in conjunction with the companies that are making them, as well as providing infrastructure for conducting those studies. And then the military arm, the Department of Defense arm, the Biomedical Advanced Research and Development Authority has a lot of experience in development of vaccines in pandemic situations and has been involved in sort of all phases of this. The group that we're actually part of is the COVID-19 Prevention Network. This was formed by the NIH, the National Institute of Allergy and Infectious Disease, or NIAID, under Tony Fauci. And the idea behind this is to respond to the COVID-19 global pandemic and to use their existing research infrastructure. And so because of this, both St. Louis University and Washington University have been involved in this network. Washington University, because of our 30 years of experience doing clinical trials in HIV, in the AIDS Clinical Trials Group network and the HIV Prevention Network that is funded by NIAID. St. Louis University is part of this network as well, as part of their 30 years of experience doing vaccine trials as part of the vaccine trial and efficacy units, the VTEUs. The idea behind the network, and it was created really to use this underlying infrastructure and considerable experience in conducting clinical trials to conduct phase 3 efficacy trials, predominantly for COVID-19 vaccines, but also for monoclonal antibodies. Part of the reason to use these networks is that they have a lot of experience in working with underrepresented minority populations and other populations so that we get fair representation in the studies and ensure that the vaccines will work and be safe for all populations involved. There are many, many vaccines in development. This is how we are able to do this more quickly, is just having a very, very robust pipeline. This is from the New York Times, has an excellent updated coronavirus vaccine tracker. You can see there are 88 vaccines that are in active development in the preclinical phase. Phase 1 are those initial safety and dosage finding vaccine studies. So can you do 1 dose? Can you do 2 doses? How much of the vaccines do you need to give and how safe they are? So those are small studies, usually maybe a couple hundred people at the most. And you can see there are 34 vaccines that are in that early phase 1 stage. Phase 2 vaccines, there are 14 vaccines in those expanded safety trials. So these are usually sort of bridging studies of between hundreds to a couple of thousand people. And they're looking again at safety in a larger population, but also do a little bit of early efficacy, mainly in terms of looking at the immune response and which vaccines, which doses give you good antibody responses. And then what we have been mostly involved in are phase 3 efficacy trials. These are large-scale efficacy studies. Most of these studies are at least 30,000 participants. They are placebo-controlled, so you get vaccine or you get placebo. There are 11 currently in large-scale efficacy studies, and currently through the CoVPN, there are 3 vaccines that are currently being studied in these phase 3 studies. There are 6 vaccines that are approved for early or limited use. That is not in the US, but there have been vaccines that are being used in China and Russia with early or limited approval. And there are no vaccines that are currently approved for full use. One of the ways to speed this up is actually to blur the lines a little bit and combine studies from phase 1 to phase 2 or phase 2 to phase 3. And so oftentimes there's an earlier phase of the study with more frequent monitoring and testing, and then it transitions fairly seamlessly into a phase 3 study. This is not something that was newly invented for these coronavirus vaccine studies. We've done combined phase studies for years. There's significant oversight to administer the vaccine studies with all the relevant safety parameters. The main oversight for this is the Food and Drug Administration, the FDA, which has very, very closely monitoring these studies. I'll talk about that in just a bit. But each individual study is also monitored by an institutional review board that is independent from the study, that examines both the initial protocol and then any safety events. The NIH is very closely involved in monitoring these studies and has its own data safety monitoring board as well. The individual companies each have their own independent data safety monitoring boards that are looking at any adverse event that would occur. And then at each individual site, there are medical monitors that are frequently on site, making sure that the vaccine studies are being conducted properly and that all relevant clinical practices are being taken. So, and we are doing these studies based on the same good clinical practices that we've been using for years that are based on the Belmont Report for ethical principles for the protection of human subjects. So there's been a lot of talk, particularly in the media, about whether or not we are going too fast. And I just wanted to give a couple of examples as to how you can go fast without sacrificing safety or efficacy. So one of the major ways to make it go faster is to put a lot of money into it. And so the US government is covering many of the costs of running the trials in addition to the industry sponsors. And the US government has guaranteed that they will purchase the vaccines. So this takes considerable amount of risk out of the companies working on these studies. And they are partnering closely with the US government to conduct the studies. So when you do most vaccine studies, usually you don't even start to plan the phase 2 trials or the phase 3 trials until the phase 1 is completed. And so because of prior data on SARS and MERS and prior vaccine work in other pandemics, such as the Ebola epidemics and influenza work, a lot of these studies, the phase 2 studies are being planned while the phase 1 studies are ongoing. So that for any of the vaccines that are passing initial standards on safety and preliminary efficacy, you can move those directly into phase 2 trials because the studies are already planned and a lot of the regulatory work is already ongoing while you are still conducting the earlier phase studies. And again, we are combining some of these phases in some of the trials. The other way to speed things up is just a numbers game. So larger studies, 30,000, 60,000 people in a study will have enough events. So the endpoint of the efficacy studies is the incidence of COVID-19 disease, and that should be higher in the group that got placebo than the group that got vaccine. So clearly the fastest way to do this would be a challenge study, but that is not considered ethical at the moment because of the mortality associated with COVID-19 and the lack of good therapeutic agents for treating COVID-19. Although there are people still talking about that. These large vaccine studies are not challenge studies. So we are relying on local epidemiology and local COVID-19 transmission that was going to happen anyways, and measuring that very, very closely. There's industry and academic partnerships between the vaccine candidates. So a lot of these are being developed by academia in partnership with industry and moving very quickly through that. The other thing is that there are multiple vaccines in development. So for most vaccines that are being developed, there are a limited number of pharmaceutical companies that are actually interested in developing that vaccine. So there are so many vaccines currently being developed for COVID-19 that you are able to go faster just because you can pick the ones that are working the best. So the populations that we want to recruit in the vaccine studies are adults currently, although there is definite interest in starting to look at doing concurrent studies in especially adolescents, but even in younger populations as well, because despite the fact that they are not being hit as hard by the disease COVID-19, they are suffering from stay-at-home orders and inability to do in-person school as well. But right now the vaccines that are being tested are all being tested in adults. The plan is to enroll at least a quarter of the population over the age of 65 in these initial studies because of the need to enroll people who are at most at risk for developing severe disease. And then to enroll people who are at risk, so beyond the elderly, also those with comorbidities that put them at higher risk of severe disease, and disadvantaged populations, including Black, Latinx, and Native American populations that have been hit the hardest by this pandemic. So why do we need to enroll all of these populations? So thus far, many of the vaccines in phase 1 and phase 2 studies were tested predominantly in young Caucasian populations. And we need fair and equitable representation to ensure that the vaccines work and they're safe in everybody. So we think they would, even if we did all of the studies in a Caucasian population, but the risks are different, the exposures are different, and the rate of progression is different. And the main risk that we take with not enrolling broadly is that the vaccines may take longer to develop because they're not being tested in the populations most likely to have endpoints and severe COVID-19, and we might miss safety or efficacy issues. So there are numerous vaccines that are in development. The earliest vaccines that became available for testing were RNA vaccines, and that's mainly just because RNA is really, really easy to make. As soon as you have the sequence for the virus, you have the sequence for the spike protein, you can develop those vaccines. There are not approved RNA vaccines that are generally available, but they have been studied in other platforms as well. And then DNA vaccines, actually the next vaccines that have become available for testing are actually vectored vaccines. These are mainly adenoviral vectors, although other vectors may be used as well. So we're using a vaccine platform then that is a different virus that is usually replication incompetent, but can present the spike protein. And then the third group of vaccines that is likely to become available for testing are recombinant protein vaccines, and then potentially inactivated vaccines or live attenuated vaccines. Those are not in late stage development yet. This is the current CoVPN planned phase 3 protocols. So the Moderna vaccine was the first one that actually got started and really didn't get started the end of July, sort of early in August got started and has just finished enrollment last week. So we are in that green enrollment phase is done, and we are moving into case accrual, looking at events. AstraZeneca was the second vaccine to get started, and that one actually did get started the end of August, but fairly quickly after it got started, it was paused due to a safety event in a participant. That safety event was very, very, very rigorously adjudicated by the FDA and other data safety monitoring boards. Every study that has been done with the AstraZeneca adenovirus platform that was being used for that, not just for SARS-CoV-2, but any vaccine with that platform was evaluated by those data safety monitoring boards. That study just came off pause last week. And in the meantime, Janssen got started, and we actually got started doing the Janssen vaccine study That also got put on a much shorter pause due to a safety event and a participant, and also got restarted last Friday. We are expecting 2 other vaccines, Novavax and Sanofi. Those are both protein-based, recombinant protein-based vaccines, and we're expecting them to start in the next month. So the 3 that are currently currently in progress by the CoVPN, Moderna was conducted at St. Louis University, and again, they have just completed their enrollment. That is an RNA-based vaccine. There are 2 doses given a month apart, and they were randomized 1-to-1 active placebo. This study was only done in the US. AstraZeneca is being done internationally. It is also 2 doses given a month apart, but the randomization is 2-to-1 active placebo. And again, that is a chimpanzee adenovirus vector-based vaccine. And then Janssen is a single dose of a human adenovirus 26. I showed you the animal data earlier, and that is again randomized 1-to-1. And both AstraZeneca and Janssen are being conducted at multiple sites around the world. So to move on from vaccination to a little bit about passive immunization, we are more familiar with this because we have been using convalescent plasma at BJC for many months now through a variety of mechanisms. It's currently available under an FDA expanded use authorization. There are ongoing randomized clinical trials to actually determine efficacy. This is using plasma from people who have recovered, who presumably have high titers of antibodies against SARS-CoV-2, and those antibodies may be protective against progression of disease, and so we're still trying to evaluate that. In the meantime, now there are several companies that are developing monoclonal antibodies, either single monoclonals or combinations of monoclonal antibodies that are being studied. We are currently conducting 2 studies with monoclonal antibodies, both in the outpatient setting. One is the study ACTIV-2, and this is currently using a monoclonal antibody from Eli Lilly in people who have early disease, so less than 7 days since diagnosis, less than 10 days of symptoms, and mild enough to still be in the outpatient setting, but symptomatic. The second is as a study in household contacts with the Regeneron antibody. So the ACTIV-2 study is actually a very complex outpatient treatment platform. So it's an adaptive platform design that compares a single placebo to multiple agents. Right now, all we are studying in that study is the Lilly monoclonal antibody. But the idea is that as other agents become available, including hopefully oral small molecule agents, not just antibody agents, that we would be able to move those into that study. And so the study has a complex design where you can move agents from phase 2 into phase 3. Agents that don't graduate to phase 3 would stop after just a phase 2 evaluation. Eligibility for the outpatient study, adults, and again, 7 days since diagnosis, or less than 7 days since diagnosis, less than 10 days since onset of symptoms, and those are sort of typical symptoms of SARS-CoV-2. And then further eligibility is kind of tailored per study agent. There are not a lot of additional eligibility criteria for the monoclonal antibodies. The study visits in the phase 2 are more frequent, but some of the major things are monitoring daily symptoms and monitoring replication of the virus in the nose and in saliva in participants. So they are seen at day 0, day 3, and day 7, but they're collecting samples and data at home and bringing those to visits. The graduation of those agents to phase 3 would be after evaluation of endpoints. The household contact study is a phase 3 study looking at the safety and efficacy of a combination of monoclonal antibodies in household contacts of people who are symptomatic. So the index case, the person who has SARS-CoV-2 must have been tested within 96 hours and be symptomatic, but not be requiring hospitalization, so still be in the household. And then the people being enrolled are household contacts of that person. Right now, it's adults. They're moving it into adolescents as well. Those household contacts have to have at least 48 hours of exposure to someone with symptomatic disease and plan on staying in the household with them. There is baseline infection status, but that does not an exclusion. So they're going to divide those cohorts and look at the data based on whether or not people were early infected and asymptomatic or not. And the agent being given is actually being given subcutaneously, so not an infusion, so a little bit easier. And then this is, again, the sort of patient journey through that study. Fairly complex with both inpatient or in-person and at-home evaluations of those folks. So I think I'm going to transition this over at this point to Dr. O'Halloran to talk to you about remaining studies.
So thank you very much, Dr. Presti, and I'm just going to try and advance the slides. So I'm going to change tack a little bit here now and talk about some of the treatments that we are currently using for the moderate and severe COVID-19 and some of the issues that we need to address in order to improve those therapeutics. So as we've come to understand, and as Dr. Presti alluded to earlier on, we're really looking at a kind of a spectrum of disease or a number of different stages in COVID-19. So we're familiar with that kind of very early infection, that viral response phase. And in reality, those are probably not the patients that we are seeing in the hospitalized setting. Over time, that in a proportion of patients, that moves on to the more pulmonic phase, where patients develop shortness of breath with hypoxia. And those are the patients that we tend to be seeing in the hospital setting. And then that can transition into the more severe patient cohort and those with ARDS and other more advanced manifestations of COVID through this hyperinflammation phase. And at present, I suppose, the therapeutics that we have are limited, but what has become standard of care for us would be three main things: remdesivir, dexamethasone, and convalescent plasma. And I think what we have seen, particularly with the studies around remdesivir, is that trying to utilize the agents at the time when they're most effective is a little bit, can be a little bit tricky and a little bit difficult. So we know from the ACT-1 study that remdesivir in a hospitalized patient population did improve the time to recovery in that population, but didn't really impact mortality in any way. And I suppose that would probably say to me that it's likely that in that situation, by the time we are using remdesivir, we may have transitioned from a disease that requires an antiviral into a disease that may require an alternative treatment. I suppose one of the things that we have seen from the studies with remdesivir are that although, and these were reiterated with Gilead's own studies, that although the mortality is not impacted, we are not seeing that there are a lot of serious adverse effects with remdesivir. So it does seem to be relatively safe, but trying to use it at an effective time is an ongoing challenge. So then that brings us to the other end of the spectrum, where we are currently using dexamethasone to target that kind of hyperinflammatory phase. And the studies on dexamethasone did show improvement in mortality. However, that was really in the more moderate to severe cohort of people. So those people who have an oxygen requirement, who are hospitalized or on mechanical ventilation. In those patients who are less unwell, it may actually have trended towards the other direction where it could potentially contribute to harm. So again, it's finding the ideal place to use these treatments. The data on dexamethasone has been replicated in a number of other studies. You can see on the top part of this table. However, so far we have not seen this translate into any efficacy with hydrocortisone or methylpred. So there is some more work still to be done on that. So that kind of brings us to the unmet need relating to therapeutics for COVID infection, and trying again to really figure out where the niche for each of these therapeutics are. So as Rachel spoke to you about Operation Warp Speed, which is the overarching program to develop vaccines and therapeutics for COVID-19, part of that is the Accelerated COVID-19 Therapeutics Intervention and Vaccines Program, or the ACTIV program. And this is a group of NIH master protocols which have been designed to look at different therapeutic options. And I'm going to just orient you a little bit to these because I think we're seeing a little bit about them in the media, and we're now doing some of these studies here. So Rachel has already spoken to you about the ACTIV-2 study, so that's the outpatient version of it. And I'm going to swing back in a moment and speak a little bit more about the ACTIV-1 protocol, which is looking at adjuvant immune modulators for the treatment of COVID-19 in addition to current standard of care. But I just want to briefly touch on ACTIV-3, 4, and 5. So you may have heard over the last week that ACTIV-3, which is an inpatient phase 3 study, a master protocol to look at neutralizing antibodies and other therapeutics, was paused and actually stopped by the DSMB, the Drug Safety Monitoring Board, and the reason for this was actually futility. At this present moment in time, the drug that was being tested in this cohort was the Lilly monoclonal antibody in the setting of moderate to severe COVID infection, so in that hospitalized cohort. And really, they found that there was no efficacy in this population. There was no safety signal, but there was no efficacy. And that again circles back to us needing to use these therapeutics at the appropriate time. So the monoclonal antibodies are far more likely to be effective in that viral phase. And now the ACTIV-3 protocol will look for alternative drugs to be entered into it and continue to test other potential antivirals in that hospitalized cohort. ACTIV-4 is an exciting one because I think this is a clinical question that many people have been asking around antithrombotics, and it's a phase 3 master protocol to look at different anticoagulation strategies in this population. And it is planned that here at WashU we will participate, and Dr. Bociccio in surgery will be the PI in this study. It's not yet open, but this will be one that we'll participate in here. And the important thing to note is that the ACTIV-1 and the ACTIV-4 protocol committees are actively working together to allow co-enrollment in both ACTIV-1 and ACTIV-4, because it is general practice that co-enrollment in more than one interventional therapeutic or an interventional clinical trial is not permitted, but in the interest of making sure that the best data is available and occurs quickly, we think that it would be really beneficial for ACTIV-1 and ACTIV-4 to allow co-enrollment without impacting on the outcomes of either of those studies. And then the final ACTIV protocol is the Big Effects trial, and that is an inpatient phase 2 master protocol. And the idea behind that one is it's doing phase 2 studies of a number of agents with a view then to being able to decide which of those studies would enter into the phase 3 studies under any of these ACTIV protocols. So circling back to ACTIV-1, and this study opened here about 2 weeks ago, and we're glad to report that it's recruiting well, and we have 9 participants currently on study. And the idea behind this protocol is it's a master protocol to look at multiple immune modulatory agents in addition to standard of care in moderate or severely ill patients. And just the definition of moderate really are those patients who are hospitalized with evidence of lung involvement on their imaging with an oxygen requirement or SATs less than 94. And as I said, it is to look at add-on therapy to current standard of care. This protocol does provide remdesivir because that is one of, or at least at the time when it was being developed, that was one of the more difficult standard of care agents to necessarily obtain. But it also allows other standard of care, such as dexamethasone and convalescent plasma. Any participant hospitalized over the age of 18 can potentially be enrolled. And the primary endpoint to this study is time to recovery by day 29, using the 8-point ordinal scale that we've all become familiar with for outcomes in COVID-19 studies. Secondary endpoints include clinical status on day 15 and day 29, and again, day 14 mortality. So I'm not going to go through all of these, but I just want to hit the high points on the inclusion criteria. And those really are that patients have laboratory confirmed COVID-19 or SARS-CoV infection within 14 days of enrollment. And in addition to that, they really need to demonstrate that they actually have ongoing illness. So radiological infiltrates, blood oxygen saturations less than 94, an oxygen requirement, or on mechanical ventilation or ECMO. The exclusion criteria for the study are not particularly stringent. I suppose two of the bigger ones are the ALT/AST greater than 5 times the upper limit of normal, and eGFR less than 30. Although there are thoughts of trying to adjust some of these parameters by the protocol committee to try and make these studies as inclusive and as representative of the populations that we are dealing with and trying to address. Again, dexamethasone and convalescent plasma are permitted. They're just not provided. Remdesivir is provided. So as I mentioned, this is a master protocol, and this master protocol allows for 5 different drugs to be trialed at any given time. And it's important to note that these are not being tested head to head. Each of these drugs is being tested against placebo. At present, within Active 1, there are 3 agents that have been selected, and I'll talk about that a little bit more. But it also allows for additional ones to be added, or the ones that are currently being used to be removed or replaced. At the moment, the drugs that are there either are given by infusion or by pill. And ultimately, the randomization with 3 agents ends up in a 1:1:1 randomization. Participants are first randomized into one of the investigational agent arms, so A, B, or C, and then within that arm, they are subsequently randomized into whether they receive the drug or placebo. So the selection of the drugs for this was a very large endeavor conducted by the NIH and a committee of experts across many fields screened about 400 agents for all of the active programs. Of those, 170 of the agents received a review using triage criteria, and then 39 of those agents were prioritized for different active protocols. There were 14 of those agents that were immune modulatory, and those were the agents that were considered for the Active 1 study, so the study that we're talking about here. And 3 drugs were selected, and those drugs were infliximab, abatacept, both of which you would be familiar with from clinical practice, and the final one, cenarivaroc, or CVC. So I'm not going to talk extensively about these drugs, but I just want to kind of highlight some of the reasons why these ones were selected. So infliximab or Remicade, which we're familiar with from the treatment of inflammatory bowel disease and rheumatoid arthritis, etc. Its target is TNF-alpha. In this particular study, it is given as a once-off infusion. And although we are all familiar with some of the safety considerations with infliximab, those tend to be considerations when it is given in its standard usage, so chronically over time for the treatment of IBD or RA. Data from the company and from other usage would indicate that actually those issues are really not large considerations in the setting of a single use. And I suppose the reason that infliximab was chosen was that some early in vivo data showed that severe COVID-19 patients had elevated levels of TNF alpha, along with some other in vivo and in vitro data. But I suppose one of the most compelling things really for infliximab's choice was probably, at least to my mind, the fact that in a number of, or in one particular large registry of inflammatory bowel disease patients, those who were being treated with infliximab and subsequently developed COVID-19 seemed to do better than their counterparts on other immune modulatory agents. And I think this is interesting, and I think we all know that the conclusion of any paper written around this would say, well, next step would be to move this into a phase 3 or clinical trial or randomized controlled trial to see is this borne out in that setting. So I think it will be very interesting to see the data around infliximab. The second agent is abatacept or Orencia, again, or this is a CTLA-4 target. And again, in this study, it is being used as a single-use agent. So just one infusion. And I suppose much of the data for the reasons behind the choice of this particular agent relate to mouse models in flu-induced pneumonia, not quite COVID-induced pneumonia, but I suppose it's one of the closer things that we have at the moment. And treatment with CTLA-4 decoupled protective immunity and immunopathological memory T-cell response, but didn't cause issues with viral clearance. And there are a number of other examples of why abatacept may be a good choice for this immune modulatory phase that we're looking at trying to address. The final drug is cenarivarok, and this is a dual CCR2 CCR5 inhibitor. It's not currently licensed for any indication, but it was originally developed as a HIV drug, and in those kind of early HIV studies, it did show safety and efficacy in HIV, but it happened to be being developed around a time when we were going through the integrase era, and although cenarivarok was shown to be effective, it's a little bit more cumbersome to use, and it just didn't really take off in that space. It is currently in phase 3 trials for NASH, fatty liver, and part of the reason that it's been chosen in this study is that patients with cytokine storm have been observed to have high levels of CCL2 and CCL5, likely caused by feedback from SARS-CoV upregulating CCR2 and CCR5, and this molecule targets that. So in the few minutes that I have remaining, I just want to touch on some of the, not so much the clinical trials that the Infectious Diseases Clinical Research Unit have been doing, but also some of the translational research that we've been supporting. So back at the start of the pandemic, in conjunction with the ECRC, Dr. Philip Mudd and Dr. Stacey House, we established WashU 350, which is a biobanking study for patients presenting with COVID-19 that some of you might be familiar with. This study has currently accrued 500 participants, so we are now following a portion of those, and already have started to see some work generated from that. At the same time, WashU 353 was established, and this was initially established by Dr. Presti and others to screen participants for plasma donation, as the blood banks did not have a mechanism in the very early days of how to go about this. Once that requirement was no longer required, we maintained the study. And this study is for people who've recovered from COVID-19 and collects biospecimens over the course of a 1-year period. Interestingly, Dr. Ali Alabidi, who's been doing much work with the samples from this particular subgroup, has recently added a bone marrow sub-study for participants who have been enrolled in this study. And in addition to that, there's also controls that are required for that. So if anyone wants to take part, all the specimens from the study are actually a university resource, and that's been managed by the ICTS. And as of, I think yesterday, over 1,500 samples had been distributed to 23 investigators. So in the interest of time, I'm going to skip over this. This is just the breakdown of the demographics of the cohort in case anybody is interested in doing anything further with this group. And just to highlight some of the work that's already been come out of sample use from this cohort, and the WashU saliva assay, among other studies. Finally, just before I finish up, Dr. Presti and I have presented a lot of the studies that are being run through the Infectious Disease Clinical Research Unit. There are other clinical trials ongoing here at Washington University with other principal investigators. And that brings me to my last slide and the opportunity to finally get to thank so many people who've been working extremely hard over the last 6 months on all of these and other studies that we've opened and closed in the meantime. I would like to particularly extend both Dr. Presti's and my appreciation to the team at the Infectious Disease Clinical Research Unit, the ID crew, in particular Michael Klebert, who we could not have opened any of these studies without him, in addition to Lisa Kessels and one of our third-year senior fellows, Adriana Rousseau, who has worked extremely hard on many of these clinical studies. In addition, I'd like to thank Dr. Powderly and Dr. Fraser, as well as Dr. Gurnett at the ICTS for their support, as well. And also, we have worked with a number of other clinical trials groups and research groups over the last couple of months, in particular the ECRC, Dr. House and Dr. Mudd, who worked with us on the WashU 350 study, as well as the teams at CCS and Trial Care, Chuck Goss at Bioinformatics, and Investigational Pharmacy and Tissue Procurement, and most of all, the participants and their families.
Thank you so much, Dr. O'Halloran and Dr. Presti, for joining us this morning and discussing all of the hard work that you have been leading here at WashU. I will go ahead and share my screen now just to give the credits, but then we can transition to the question and answer portion for the last couple of minutes here. We can get started with one question that came in. So this question asks, why did the investigators of the Active-1 study choose a 2-week mortality if patients with severe COVID-19 tend to have longer hospital courses?
So that's one of the secondary endpoints. The primary endpoint is 29 days, and there is also other secondary endpoints that go out to 2 months.
Okay, thank you for that. Another question that came in that Dr. Presti was able to type in an answer for the person who asked it at least. So it asks that given the primary outcome to evaluate the efficacy of vaccines is dependent on the incidence of COVID-19 infections, how do researchers account for the dichotomous population of individuals who are socially isolating and those who aren't? If the trial primarily recruits individuals who are correctly socially isolating and wearing masks, does that not artificially make their outcomes appear better?
Right, and so I did kind of put in an answer there. So we are definitely trying to enroll people who are at risk of both infection and of severe disease. That is all for the most part individual report. I think if we talk to somebody who wanted to participate in the study, but had not left their house in the past 6 months, and was having food and everything delivered, and was able to rigorously follow social isolation and mask wearing, they would not be a good candidate for the study. So we're basing this in part on evaluation of demographics, of evaluation of people's underlying risk for infection based on their occupation or other social characteristics, such as living in a multi-generational household with some of the family members or members in that household needing to work outside the house and therefore being put at risk. So while there are not specific requirements delineated in the protocol does require that in the assessment of the investigators, these people are the participants are at risk for acquiring SARS-CoV-2 infection.
Great.
I hope that's helpful.
I think that was a kind of a question that popped up in my head too, and that seems like the most pragmatic way to go about that conundrum. Of course, I mean, we ourselves as physicians are, not just in our homes all day, although we are wearing masks and doing all the correct hand hygiene and social distancing as possible. So that was an interesting question. Thank you for whoever submitted that one. And then just one more quick question for the sake of time. This is coming from one of the geriatricians. Have any of the vaccine trials enrolled patients in long-term care facilities?
I guess it depends what you mean by long-term care facilities. The studies are somewhat intensive and require sort of the ability to check in via electronic means twice a week, which does make it slightly more difficult. So they're not specifically targeting people in long-term care facilities, but if they are able to get to the study site, or if they are able to participate in the study, there's no exclusion for people living in community settings or long-term care facilities. So we would love to have them in the study. It's a balance between them being able to actually participate fully in all the requirements of the study. They're not excluded, and we would be happy to have people involved.
Okay, well, thank you again to the two of you for joining us this morning. I think that's all the time we have for questions for the sake of time, but feel free to reach out to me. My name's Ava Kadiwara if any other questions come along, and I can send them to Dr. Presti and Dr. O'Halloran as well.